Hello, everyone. Thanks for being here. I'm going to describe to you one of the projects I worked on this summer in which I screened integration lines, Slim Citian's integration lines for shelf life phenotypes. I'm sure we all know the importance of tomatoes as a fruit crop, or plenty of crops. But tomatoes face large losses, like post-harvest losses, and even sitting on the shelf. Many fleshy fruits do. And we see here's a metric: nearly 45 to 55% of all fruits are lost during post-harvest handling and storage. So this isn't even limited to just tomatoes. And this third metric here, nearly a third of the U.S. food supply is lost, after they're harvested. So, working in both a tomato biology lab and tomato breeding lab this summer, it was of interest of us to find, sources of genetic variation that might in the future confer extended shelf life for tomatoes, so that we can, get these products to consumers before they go bad. And luckily, we can turn to distant relatives of tomato, the nightshade family, to find these sources of genetic variation. So in this study, I was looking at Solanum sitiens, which is a nightshade endemic to the Atacama region of Chile. And these tomatoes are super interesting. I kind of coined them the super tomato here. The Atacama region is a very arid place, so this tomato is highly tolerant to salt, drought, and cold temperatures. And recently, there has been an introgression line library established for this tomato. And before I get into that, I should explain what integration line even is. So an integration library is established by crossing two individuals. So in this case, a Solanum sitiens tomato was crossed with a Solanum lycopersicum tomato, which is this domestic tomato. And when you repeatedly backcross the F1 progeny with one of the parents and then inbreed them, you can create a library that spans these single integration segments, chromosomal segments that come from the wild line, and then it's all in the background of domestic tomato. And then you can study all of these lines separately to see what chromosomal chunks confer specific phenotypes from the wild line. And that's just what I did. So recently, the Tomato Genome Resource Center put out these integration lines for Solanum sitiens. It's one of the newer lines, and our labs had the opportunity to get their hands on these lines and study them. And so the goal of this research was to record and characterize phenotypes associated with these lines, more broadly, under the umbrella of characterizing these lines themselves. Right? We don't know a lot about the different phenotypes these lines might confer, and they can trace back to the wild relative. And then kind of in a subset of that, I was screening for extended shelf life properties in some of these lines. And the way I did this was measuring the weight loss, which is also the water loss of these fruits over time. And by proxy, this is also a measure of their cuticular conductance, which I just learned. Because, when fruits lose water over time, they have reduced turgor in the cells, and they are, more susceptible to pathogens. And so they rot faster. They look, unappetizing when they're sitting on shelves because they're starting to wrinkle. They're losing their water. And we see here 5 to 10% water loss can render fruit completely unmarketable. So while I was tracking the water loss of these tomatoes over time, I also was grading them via some USDA standards, which I will get into later. And then I took a lot of photos of these tomatoes over the course of this summer, which we were phenotyping them from as well. So just a lot of photo sessioning went into this. And as a caveat, just informationally, these tomato fruits were supplied from the Vogel Lab salt trials. So while this is just a screen of the integration lines themselves, I also had the opportunity to look at a little bit of an abiotic stress component, seeing the difference in how salt is affecting these tomato lines versus non-salted integration lines. However, this is not a salt stress experiment, so know that. And to backtrack a little bit, I come in at the second screening of these integration lines. So this past winter, a different student in the lab screened these integration lines coming from the field trial. And they found that, the preliminary findings were that there was greater water loss in these salt stress fruits relative to the non-salt stress fruits. So here's just a simple plot, and the positive or the plus signs here are where the fruits are salt stressed within a single line. So 5265 here is salt stressed, this one is not. And we see there's great variation in the amount of water these tomatoes are losing. And this plot here is showing us that while some lines were losing much more water after they had been salt stressed, there was one line that emerged to be salt tolerant, right? And Greg Vogel's trials did determine this line to be salt tolerant, but that's less of an interest to us because at the end of this period, we saw even without this like salt stress story, there's a great difference in how these fruits were persisting on the shelf over time. At the end of this period, we cut into these tomatoes, or they cut into these tomatoes, and we see that this domestic tomato here has, a ton of precocious germination. There's like some dry rotting and wrinkling of the skin. And after that same amount of time, they also cut into this integration line right here, 5265, and it's still very well formed and marketable, maybe even edible after all this time. However, we couldn't make really strong predictions or, couldn't like take away a lot from the study because there was such like small yield of fruit. Most of them were lost to blossom and rot. So that's why we have trial 2, which is where I come in for the screening. And in this trial, we had much more fruit to work with, and we saw quite the opposite of what we saw in this first trial, such that salt stress was really not impacting the shelf life of these fruits at all. And this was with like much more fruit to actually work with. So this is, much more rigorous than the first time around. So even though like the salt component's not there, what we do see is that there is large differences in the amount of water being lost between the lines themselves, whether or not they're salted. So for these controls, there's not much difference in how much water these two lines are losing, but this line here is losing very little water over time. And this is the difference between 3 weeks and then more closer to 8 weeks. And I plotted the controls of all these lines. So, salts out. And I just wanted to see the trend of how much water was being lost by these lines on the shelves over time. And we see this line 5265, which emerged as our line of interest even in the first trial, is losing such little water over time. This is a 3-month period, and it hasn't even lost, just over 10% of all of its relative weight. And then we compare that to the domestic parental line right here, which has lost much more water over time. And these lines end when more than half of the population within that genotype was lost to rotting. So that kind of gives you an idea of how, long these fruits can actually persist on the shelves. And this is just what that line looks like right here. It has a chromosomal segment from the wild population or the wild species on the short arm of chromosome 5. And then to complement this, I scored these fruits to see if, actually if the loss of water is being attributed to a reduction in quality over time. So I threw together this little infographic, if you would, where I combined USDA processing standards for the grades of fresh tomatoes with consumer standards for the grades of fresh tomatoes. And very plainly, it's when you move from right to left, that's just a reduction in quality. The pericarp is becoming softer to a point where it's not marketable anymore. There's more wrinkling and discoloration, and then eventually you get to here where the fruit is coming to rot. And just as a note, there's like a whole area of fruit that like looks way worse than just this section. But I wanted to look very specifically within like the upper portion of fruit decay over time, if you will. So anything that was like this and worse was just kind of like scrapped as a 0. And so when I graded these tomato lines, the lines that actually had enough tomatoes to grade, we also see that it's validating this story where LA 5265 is losing less water over time and thus staying, of high quality, right? After 3 weeks here, we see that most of these tomatoes are still in that 5 range, right? And it's such a small, area because like they're all hitting that 5. And then we have some variable qualities between these other lines, and there's a large difference between this and the parent. And then after 54 days, same story. This 5265 line is still staying strong on the shelves, whereas like everything is dropping off over time, especially compared to that domestic parent. And like I said, was also just like a phenotypic screen in itself. So kind of moving away from even the shelf life story, I thought it would be interesting to show you guys some of the different phenotypes that these lines actually were presenting over. So these for day 14 and day 79 within these integressions. So like what's something's interesting about LA5252, this line that has a very short chromosomal segment in the middle of chromosome 2, this tomato, the control of this tomato didn't produce any fruit, but the salt-stressed plant produced plenty of fruit, and it persisted on the shelf pretty well. So it's showing us that perhaps, if we were to look greater functionally or genetically into what's going on in this short segment, like maybe there's some sort of positive association with the salt stress that's actually allowing it to fruit. Here, LA5254, it has a large segment in chromosome 3, and we see that there is some carotenoid inhibition here, and that is very likely due to some sort of mutation in the PSY1 gene, which is part of the carotenoid pathway, right? And we have some preliminary hypotheses that this carotenoid inhibition is also a reduction, or is aiding in a reduction of shelf life, because these tomatoes rotted much faster than any other tomatoes that actually produced reg pigments. Another one, LA5262, is just fun because it looks really crazy, even coming straight out of the field, and has just extreme reticulation and suberization of the fruit epidermis. And then, of course, LA5265, which is kind of the darling sweetheart of the screen itself. And taking a closer look at that, yeah, here we go. 5265. This is after the whole screening period. 3 months is up. This is day 86, after, like, right before we shucked all these. They're rotting on the shelves. We want to get rid of them. Look how good these tomatoes look. Would you buy these in the store? Maybe. They don't taste good. They taste horrible. They taste horrible. But they look great. And, for comparison to the domestic parent here, we have, some variation in how far along the, rotted these are, or moldy. But, these tomatoes are still, by this day, had a firm pericarp. They look good. There's no even like imprintation on like where, on where they were sitting. Like there's no wrinkling at all. And it's interesting to think like mechanistically, like what might be, genetically, what might be conferring this extended shelf life, which is part of what this whole project aims to do, right? Like when we're collecting phenotypic data on these lines and we can pair that with the actual mapping of these integration lines, which is to come, we can do quantitative trait loci mapping and analysis, which is part of like the future direction of this project. And then some other things of note is we hope to in the future rescreen these lines of interest with the complete integration library. I was only able to look at like half of this library because there's like germination issues or like fruiting issues in the field. So there's like a whole bunch of lines in this 56 line library that's worth looking at. And then, we hope that this can, lines of interest, especially LA5265, can actually be introduced into like a breeding program so that like we can get this out there and we can keep our fruit from going bad on the shelves. Funnily enough, 5265 did get introduced into a breeding program here on campus, but for its salt stress, that was before that we saw that it had these shelf life extension properties. So it would be interesting to see if it confers both of those, surprisingly. And yeah, that's about it. I would like to thank my mentor, Lance, and Dr. Giovannoni for their mentorship and help on this project. And then also Dr. Vogel, I'm not sure who's here. And of course, Delaney, Megan, and George for making this program possible. Huge shout out to Aaliyah. We were like thick as thieves this summer working on each other's projects together. All the funding sources, and yeah, thank you. Yes, one of you. I have no idea. It has like a grassy taste, which if I would refer you to talk to my mentor after this, he has some ideas of like chemically what might be giving it that taste, but no, I have no idea. Another aspect could be like it just sat on the shelf so long. Like we tried these fruits on like the back end of the trial, so maybe it lost flavor over time. Yeah.
How did you make sure that like ethylene production or some fat hormone that releases the fat hormone when it's rotting, that it did not affect the wrapping of the other people? Create like these clusters when the adjacent tomatoes around the rotten tomatoes also ripen faster.
You mean like a group effect where like they're releasing ethylene and affecting others around them? There was no control for that really. Yeah.
Like, did you have like each category group and like adjacent to each other, or did you scatter everything?
They were all scattered. Yeah, yeah, yeah. So in that sense, yes, there was no like group effects within lines, but there was no measure of ethylene. Yes. Oh, I mean, that would be like a shot in the dark. I really couldn't say. There's, it has quite a large chromosomal segment coming from the wild Cytians. So there's like a whole slew of genes that can be contributing both to its extended shelf life and its flavor. So sorry, I don't have a good answer for you on that.
So other integration lines that have that same segment or parts of that integration?
Yeah, so these integration lines have like the integrations themselves are overlapping. So yes, but the lines that were overlapping surrounding the line of interest didn't show any of the same attributes phenotypically. Yes. A long time. I mean, just because, we have like handwritten notes of these tomatoes, like when we're actually like observing them phenotypically, like over time, but also like we need to make note of like this one has, dry rot, this one has this mold. But there was like so many tomatoes to screen that we also paired that with like photographs. So when you could couple those together, but I would say like the grading took less time, more so than just like organizing the photos, like organizing them by name and then grading those. So it was kind of like a whole situation, but yeah, it was a good time. Yes.
I don't know if you were talking about this already, but question about the taste. You're saying that like in the future, I think you're gonna be like tie this into like making the shelf life more extended for those crops. Do you foresee like a problem that if the taste is bad, that the shelf life is still won't want to buy them because they taste bad? Is there some sort of direction like looking at the taste again?
Yeah, I mean, we didn't touch anything on taste, but the idea would be to incorporate that like integration segment, the genomic segment coming from Cytians that's conferring this extended shelf life and being bred with other attributes of tomato that would give it that taste. So yeah, surely this line would need some support from other genetic areas to make them taste good. And be able to persist on the shelves.
One more? Okay, one more question.
You mentioned initially about the super tomatoes coming from the Atacama region.
Yes.
in Chile and the area around. Do the people who live in that region eat those tomatoes or no? So
Yeah, interestingly, those tomatoes never ripen. Part of their reproductive strategy under stress is to desiccate, and then to regrow from the ground. So yeah, the tomatoes never like fully form. That picture that was like they were green, they just look like that.
good forever.
So yeah,
Okay, thank you.
yeah.
So do those Atacama tomatoes, so do those also have a similar grassy taste then, since they never ripen?
Presumably, I haven't tried them, so I don't know. Thank you.